We're talking about RET fusions here, and fusions, as we know, are the same as chromosomal rearrangements. These are genomic alterations. These are findings in a subset of non-small cell lung cancer tumors. It is rare to find some of these, but they're very important. When we talk about these types of genetic changes, these are acquired. And by that, we mean they're not present at birth. They're not things that we pass on to our offspring. It's unclear really why these occur. And we're still in the process of understanding why these fusions develop in some people, but not others. Identifying these fusions when they're present, though, is critical to care. It tells us a little bit about the cancer. It explains the biology, but it's much more than an academic exercise. What these fusions tell us is really how to attack the cancer. It exposes these unique vulnerabilities to these cancer cells, and it guides our treatment right from the beginning. It guides us towards targeted therapy. It steers us away from ineffective immunotherapy. But individual tests for these fusions is impractical, is very difficult. In the early days of targeted therapy, we would do tests like FISH for ALK and for ROS1, but our list of relevant drivers increases and stretches past a dozen. We can't do all of those tests. It's inefficient. It uses up a lot of tissue. It's actually more expensive. Really, in the modern days, next generation sequencing is our best approach. But the type of sequencing is also relevant. And I want to make a quick plug for the potential added value of RNA sequencing. When we talk about next generation sequencing in the US, that's primarily using DNA as the nucleic acid. And this is a good test. It's very good at detecting certain things, such as EGFR, very easy to find on DNA-based next generation sequencing. But RNA sequencing adds a dimension which in some cases is useful. Not for everyone, and certainly it does add cost, it does add time and expertise, but there can be value in finding these alterations. This is a study that was released from Dr. Raima Benayyed when she was at Memorial Sloan Kettering, and they took patients with advanced non-small cell lung cancer. All of them got DNA-based next-gen sequencing, and if they found a driver, they moved over. But those that did not have a driver using DNA NGS, there were 254 of those. They sent those for RNA sequencing. And out of those 254, they found an actionable driver 13% of the time. 13% is not a small number. And what did they find? They found things like ROS1. That was the most common one that they found. ALK, NRG1, NTRK, RET, the things we'll talk about now. If we would have missed these alterations, these patients would have gotten the wrong treatment, because there really is a right and wrong treatment in many of these cases. If we miss those drivers, patients are treating the wrong way. And so the stakes are really high. If we don't find anything by DNA-based NGS, we need to start thinking of RNA sequencing as a way to really properly and thoroughly test for these alterations. So let's talk about RET. RET stands for rearrangement during transfection, RET. This is a receptor tyrosine kinase protein, and it has a normal function. So this protein plays a role in our development. It's important for cell growth. It's supposed to do that. But generally, when we're in development, not as adults, when there's too much RET signaling or inappropriate RET signaling, when RET is turned on, when it's not supposed to be turned on, that causes uncontrolled growth. That leads to cancer. And because of that feature, RET, the gene RET, is referred to as a proto-oncogene. Now, RET mutations are associated with cancer syndromes. RET mutations are things that we inherit and can pass on. They're associated more with thyroid cancers and syndromes called multiple endocrine neoplasia. Now, that's not what we're talking about for lung cancer. For lung cancer, we're talking about RET fusions, RET rearrangements. These were first described in the laboratory in 1985, first seen in a patient's tumor in 1990, but the first RET fusion in lung cancer was described in 2012. So we have about 10 years, which is relatively short history, but we've come a long way. When we first started to see RET fusions were present in lung cancer, we knew that there were drugs out there that were active against RET. RET's not common. These fusions are present in about 1 to 2% of lung cancer. There are different fusion partners. When we talk about a fusion, we're talking about two genes coming together. The RET is providing the kinase portion. The other gene, the fusion partner, is most frequently KIF5B, though we also see CCDC6, NCOA4. These letters probably don't carry much meaning today. With some of the older drugs, they actually did have some input into how well those medicines worked. with the current drugs. They don't seem to matter too much. They may be important in the future. For now, all that's relevant is that RET fusion portion. And we have a lot of drugs off the shelf that are available, that are approved for other cancers, such as kidney cancer, thyroid cancer. These medicines do block RET signaling, but they do a lot of other stuff as well. They're multi-kinase inhibitors. They're what we called dirty kinases in the past, where they will block RET, but you're also going to get blockage of a lot of other things too. Now, blocking those other proteins might be important for kidney cancer, for thyroid cancer, not so much for RET cancer, but they all come together. It's part and parcel. So when we looked at these older multi-kinase inhibitors, they worked. The response rate probably around 20%, but they had a lot of side effects. And those side effects were the result of blocking other proteins. RET inhibitors were listed in the NCCN guidelines, these multi-kinase inhibitors, as options to consider, but they weren't frequently used, again, because of toxicity. What we really wanted was a selective RET inhibitor, something that could block RET signaling, but not block a lot of other things. If we could design a selective RET inhibitor, there's not much consequence for shutting off RET signaling when we're an adult. So we can live without RET as an adult. If we completely block RET and don't block anything else, we should have a drug that works very well, that works for a long time, and that doesn't have as many side effects. And so we relied on our chemists, our medicinal chemists, to design these drugs, and that's what they did. There are two FDA-approved selective RET inhibitors, Selpercatinib, previously LOXO-292, and Pralsetinib, previously BLU-667. These are both approved in 2020. They are oral agents. They work well through the body and through the brain, and they're both very well tolerated. Both agents represent current standards of care for the treatment of RET fusion-positive non-small cell lung cancer.